Good day. I am Brian Hoffland, President of Research to Prevent Blindness. Welcome to RPB's Lunch and Learn, Eye on Retinitis Pigmentosa. Research to Prevent Blindness is the leading nonprofit organization supporting eye research directed at the prevention, treatment, or eradication of all diseases that damage and destroy sight. RPB was founded in 1960, and over our 62-year history, we have awarded more than $397 million in research grants to the most talented vision scientists at the nation's leading medical schools. As a result, RPB has been associated with nearly every major breakthrough in the understanding and treatment of vision loss. In today's educational session, you will hear from two RPB-supported talented scientists about retinitis pigmentosa and its causes, impact, and cutting-edge research. One of our speakers, Dr. Jackie Duncan, received an RPB Career Development Award and an RPB Nelson Trust Award for retinitis pigmentosa. And the other, Dr. Krzysztof Palczewski, received an RPB Stein Professorship Award and an RPB Stein Innovation Award. He also served in RPB's Scientific Advisory Panel for many years, before and then after his RPB awards. Thanks to all of the generous supporters of RPB who make our work possible. I'd especially like to thank Janssen Pharmaceutical Companies of Johnson & Johnson for their support of today's educational event. The importance of supporting medical research has never been so apparent. We thank all of you for your partnership in supporting innovative vision research to ensure that new breakthroughs are possible. If you have not yet supported RPB, please consider doing so to help make sure we are able to continue with these breakthroughs. But getting back to today's session, let me introduce our moderator. Sheila Hickson-Curran, Doctor of Optometry, FAAO, is Medical Affairs Leader of Retinal Diseases at Janssen Pharmaceutical Companies of Johnson & Johnson, which works to create a future where disease is a thing of the past. They fight sickness with science, improve access with ingenuity, and heal hopelessness with heart. Dr. Hickson-Curran has worked for Johnson & Johnson and its related companies in many leadership roles since 1995. An optometrist by training, Dr. Hickson-Curran currently focuses on bringing gene therapy solutions to market for previously untreatable inherited retinal diseases and other ophthalmological disorders. Prior to her roles with J&J, she served as a clinical optometrist and conducted clinical research in education. Dr. Hickson-Curran will introduce our two speakers for today and moderate the Q&A session at the end. Each speaker will speak for 20 minutes and I will serve as timekeeper. Dr. Hickson-Curran, take it away.
Hello, everyone. Thanks for attending today. We're thrilled to be sponsors of this event. Our event today is on retinitis pigmentosa, or RP, a group of rare genetic eye diseases that affect the retina, causing vision loss and, for many, legal blindness. This topic is important because retinitis pigmentosa makes up about half of all inherited retinal diseases, or IRDs, also sometimes described as inherited retinal dystrophies or inherited retinal degenerations. So as a group, retinitis pigmentosa affects somewhere around 90,000 individuals in the USA alone. And as a progressive disorder leading to legal blindness with essentially no treatment for the vast majority of people, it can devastate lives and families. These conditions are especially important to Janssen Pharmaceuticals because we're on a mission to restore and preserve vision in patients with blinding retinal disorders by developing transformational therapies that have an advantage over standard of care. And in the case of RP, as mentioned, for most, there is no care or no treatment. Although we don't have any approved ophthalmology products at the moment for these diseases, we have active clinical programs in X-linked retinitis pigmentosa, achromatopsia, geographic atrophy, or the late-stage form of dry age-related macular degeneration, and in diabetic eye disease. Now I'm pleased to introduce our first speaker, Dr. Jackie Duncan. Dr. Duncan is exceptionally well qualified to give this presentation today. She's currently Professor of Clinical Ophthalmology and Interim Chair at the Department of Ophthalmology and the Academic Director of Retina Service at the University of California, San Francisco, UCSF. She completed medical school, internship, and ophthalmology residency at UCSF and completed a medical retina fellowship at the University of Pennsylvania, where she focused on patients with age-related macular degeneration and inherited retinal diseases. She has expertise in the diagnosis of these patients, and she focuses on, as we mentioned, AMD, retinitis pigmentosa, cone rod dystrophy, and Stargardt's disease. She's received a lot of research funding, including Research to Prevent Blindness, as Brian mentioned, the Foundation Fighting Blindness, the National Eye Institute, and the US Food and Drug Administration Office of Orphan Products. So as such, she's a perfect person to be giving you this presentation today. Take it away, please, Dr. Duncan.
Thank you, Sheila. And I'd like to start by thanking Brian and Diane for inviting me to speak with you today. It's really an honor to speak and teach you a little bit about something I'm very passionate about, inherited retinal degeneration. I'm going to also use this as an introduction to some of the research we have been undertaking with support from Research to Prevent Blindness and other sources to help us better understand how these diseases affect vision. All the work on the research I'm going to be presenting to you today has been performed in collaboration with two brilliant scientists that I work closely with at the University of California, Berkeley, Dr. Austin Roorda, and at the Medical College of Wisconsin, Joe Carroll. I would like to provide you with a list of my financial disclosures, all of which I recused myself from January 1st of this year, where I was serving as an advisor or received grant support from some companies that are conducting clinical trials in patients with retinal degenerations. So to begin, the retina is a part of the eye that serves a little bit, if the eye is like a camera, the retina is a little bit like the film. The retina is the part of the eye that takes light and turns it into a message or a picture that your brain can interpret and understand if it's working properly. So here are some parts of the retina that we'll talk about today. This is the optic nerve that takes the information from the eye to the brain. These are blood vessels that bring oxygen to the retina so that the film can take a picture of the world. And this central part of the retina, I've shown with a white square, is the center of the macula where the fovea exists. This is the very center where there's no blood vessels and only cone vision cells to allow the very most sharp and high detail vision to occur. So there's two broad categories of vision cells. And the vision cells that I'm going to talk about today are the photoreceptors. The two categories are rods, which help us with night vision and some of our side vision. So they're helpful for nighttime. And cones. There's three different kinds of cones, and these are good for fine detail vision and color vision. And they're the only kind of vision cell that exists in that central region, the fovea. So sometimes the rods and the cones don't work properly because of a genetic disease that causes them not to work properly and eventually to die. The first one I'm going to speak about today, the one we'll speak about most today, is retinitis pigmentosa. Retinitis pigmentosa is a description of a very broad category of diseases, all of which share in common death of the vision cells. In retinitis pigmentosa, which is sometimes called rod cone dystrophy, the first cells that are affected are the nighttime vision cells, or the rods. First, the rods don't work, causing night vision loss, and then they disappear and die with subsequent effects on the cones. So eventually the side vision doesn't perform as it should, and then eventually patients lose their central vision. This is an inherited disease which runs in families. Sometimes, and most commonly perhaps, it is inherited in a way called autosomal recessive, meaning you need two changed versions of a gene, one from each parent, to have a vision problem. And for that reason, sometimes only one person in the whole family is affected. It can also be inherited in a dominant manner, which means that usually one of the two parents also had the disease, because you only need a copy from one parent to have a vision problem. It can be inherited as an X-linked condition where the mother is a carrier, sometimes affected and sometimes not, and boys or men are the most severely affected. And in fact, it can also be inherited by caused by mutations in the mitochondria, which are a little different from the nuclear chain DNA that we talk about all the time. And it turns out that more than 300 different genetic changes can cause retinal diseases, and over 80 of them can cause retinitis pigmentosa. But to date, very few treatments exist, as Sheila was saying, for any of the inherited retinal degenerations, including retinitis pigmentosa. Well, when your doctor takes a look at your eyes, to let the doctor know that you have retinitis pigmentosa or rod cone dystrophy, some of the features of this disease include complaints that you might have or symptoms, such as difficulty with night vision, difficulty adjusting from the light into the dark, we call that dark adaptation, trouble with side vision, and this is a visual field test showing that the center sees very well, shown in white, and there's ring of dark area where the side vision is reduced. And the central vision, as shown in this visual field test, is well preserved until late stages of disease. This is because the central vision is performed by the cones, and the cones are not the primary site of disease in retinitis pigmentosa. They are only affected secondarily after the rods have gone away. When the eye doctor looks at your eyes, some things that the doctor might see include some debris or inflammation behind the lens. The blood vessels can look very thin. The optic nerves can look kind of pale. And there are features that sometimes the doctor will describe called bone spicules, which are just pigmentary changes, which I'll show you in a minute. In addition, sometimes the doctor will say the patient has cataracts, and that is shown in this slide here as a dark area in the center of the lens. That's a feature that sometimes reduces the central vision at early stages, even when the cones are well preserved and not affected. So when people have retinitis pigmentosa, this is a histological picture of the retina in side view or cross section. And the way it occurs is that the nighttime vision cells, or the rods, first lose their outer segments. This is the part of the rod that turns light into a message the brain can understand. Then the inner segments and then the nuclei go away. And this is a picture showing the inner segment, the nuclei up here, the inner segments here, and the outer segments of a normal eye going away in a animal model, actually, of retinitis. This is actually human retina with retinitis pigmentosa has lost all of the outer segments. There are a few remaining nuclei, and the inner segments are very compressed. But you can see the retina is very thin after a patient has had retinitis pigmentosa with degeneration of the photoreceptors. And the important thing to know is that before the rods and cones disappear in a structural way that we can see on histology, they don't work properly. So people lose their function before the structure is lost. It would be important to identify remaining cells that aren't working properly before they've gone completely away, because those might be excellent targets for treatments. We have a tool in the clinic that we use every day, which gives us pictures of the retina in side view, kind of like that histology slide I showed you a minute ago. This is the picture your doctor can take in the clinic that doesn't take almost any time at all, doesn't involve any injections, doesn't hurt. It's called an optical coherence tomography picture, which is a side view of the retina. Looks a lot like the histology slides. And I'd like you to focus your attention on some bands. It looks like a 10-layer cake, actually, but the bands that we want to focus our attention on today are this dark band. The outer nuclear layer is where the cell bodies of the vision cells reside. This little faint bright band is the external limiting membrane, which separates the cell bodies from the inner segments of those vision cells. This very bright band is a connection between the inner and outer segments of the vision cells. And then this band down here that's bright is the connection between the photoreceptors and the RPE cells. And each of these gives us information when patients have retinitis pigmentosa. So here's an example of an OCT picture in a patient who has retinitis pigmentosa. Here's someone who has good central vision, but has lost the vision off to the side. And here we see in the center, the retinal layers that I showed you a minute ago are almost normal. But as you get further away in the area where the vision is lost, you lose some of these bands. Some of the bands we just talked about become very thin, and then they go away. So we use this tool to look at the retina in patients with retinal degenerations in the clinic. Here's an example of a patient with retinitis pigmentosa that you can tell has retinitis pigmentosa based on the bone spicule pigmentary changes, the disc pallor, and then a pretty well-preserved area of retina in the center. But this patient has very severe central vision loss, and you can see her visual field shows very little vision in the center, but good vision way off to the side. Why would that be? Well, this is a patient who actually has lost all the outer segments, which is where the function of the vision cells takes place, but she still has some remaining cell bodies at the center of the retina, suggesting that maybe these cells, if they were given a treatment, might be responsive to the treatment, and we might be able to restore some vision to this patient. So our goal is to try and identify cells that haven't gone completely away, which might be good candidates for treatments. So what we really need is better ways of looking at the vision cells in patients with retinal degenerations as they're undergoing degeneration. This will allow us to identify the patients that might be the best candidates for clinical trials, who might be most likely to respond to the treatments that we're trying to develop. This might let us determine whether the treatment is helping in shorter periods of time. But this is hard because even if I squint very, very hard, if I look closely at a retinal picture, I can't see those individual vision cells because they're clear. So we use some special techniques called adaptive optics to help us see the vision cells more clearly. And when we dilate the pupil, it helps us allow more light to come in and out of the eye. But the problem is that even the healthiest of eyes have little irregularities, very subtle irregularities in the cornea and lens that prevent us from seeing the individual vision cells with clear views or high resolution. So there's a technique called adaptive optics that measures how the light is being bent by these subtle irregularities in even healthy eyes and corrects or compensates for those aberrations such that we can have very sharp images of the retina using a special approach shown here. So what this technique does is to overcome blur introduced by optical aberrations in even the healthiest of eyes using a special wavefront sensor that communicates with a mirror, which changes its shape to compensate for these irregularities. So we get a very high resolution image of the retinal vision cells. This is a picture of the machine that we use in collaboration with my colleague, Austin Rorda at UC Berkeley and Yuhua Zhang, who is now at UCLA. This is a machine that does the technique that I was just describing. And here's the result of the addition of adaptive optics. If we look at a picture of a retina, we don't really see vision cells. And if we look even with the special machine to see the vision cells clearly, unless we correct for those aberrations, we can't see individual vision cells. But if we add adaptive optics, which I'll show you here, we turn on the adaptive optics, we begin to see little white spots amongst the shadows, which are created by the blood vessels. So these little white spots are each light being waveguided by a single cone photoreceptor. So each of these little dots represents a vision cell that is able to see in this patient. And we can tell exactly where it came from because of the dark shadows, which line up perfectly with the blood vessels that were from the same location that the image was collected. We can further refine this technique by collecting not only the light that comes through a small pinhole, which is called a confocal image, showing you the kind of images I showed you before, but if we also collect the scattered light that doesn't go through the pinhole, we get an image called a split detector image. And what's helpful and powerful about putting these two together is the confocal images show us cones that have intact inner and outer segments. Those are the ones that are most well equipped to reflect the light. But if we use that in combination with the light that is not going through the pinhole, not focused in the same plane, we can see cells that have lost some of those inner and outer segments, which remain. They're still very much present, but not working properly. And these are the kinds of cells that we are most likely to be able to target with potential new therapies. And I showed you before the optical coherence tomography picture in side view. Well, the images I just showed you from the adaptive optics correspond to these same regions. This comes from the inner segments. The split detector images come from the inner segments, whereas the confocal images come from the cells that have intact outer segments in touch with the RPB cells. So you may say, well, that's lovely. I'm very happy to hear that the vision cells exist, but I don't really care about them unless they can see. So you may be asking the right question, which is, what about visual function? So, and I said before that visual function is abnormal even before the photoreceptors go away. So even though it's important to image photoreceptor structure, that only matters because it predicts how well a patient can see. So most people, including regulatory agencies, care a lot about how the photoreceptor images we collect correspond to vision. So we need to not only assess the structure of the vision cells, but also test how well they can see or respond to light in patients with retinal degenerations. We can turn the adaptive optics technique I showed you before into a way of precisely targeting those vision cells with small flashes of light. So we can ask the patient how well they can see on a very localized basis. So here, when we do adaptive optics, the patient looks into the machine and sees a red square, which we can administer a little green flash of light. And with special fundus tracking, we can deliver the same flash of light to the same group of cones repeatedly over time by tracking the blood vessels and the cone features. So we can tell exactly what this little group of cones is doing by asking the patient what they see when they see these different levels of light. This allows us to test the vision of the cells that we're measuring, we're visualizing. And we can get a good idea about how well the cell structure can turn into function that will help benefit their patients. So there are some patients that have cones that can actually see, but we don't picture. This is the sort of flip side of what I said before, where people have no vision but still have remaining cells. There are also situations where patients have cells that we can't see, but they can still retain some function. And this is a project that was done in collaboration with my colleague, Jonathan Horton, here at UCSF. This is a patient that we saw who had flashing lights in her left eye, young woman, for the past several weeks. And we did a visual field test, which showed this dark area, indicating she wasn't able to see off to the left side of her left eye for many weeks. This is what her retina looked like. It looked entirely normal. Here's her optic nerve, and her retinal blood vessels and the fovea all looked fine, no matter which different kind of pictures we took. And what's interesting, though, is that the OCT scan I showed you before showed retinal loss of reflectivity. The cell layers were all there, but they were less bright in this area that corresponded to the vision loss. And if I magnify that image here for you on this slide, you can see that the bright bands that we see on the normal eye, which has normal vision, are very dim. They're still there, but they're not very reflective. So we decided to study this region over time and look at the cones in this area. And similar to the OCT scan, we found that the cones in this area were very dim. And in fact, it looked like they weren't there at all. And that actually we were seeing the underlying cells, the RPE cells in this area where her vision was reduced. This is in comparison to the same location in the other eye where we saw lots of cones that were working properly and still present. So in comparison, we saw what looked like a loss of cones. Well, this patient had an inflammatory disease that got better over time called acute idiopathic blind spot enlargement syndrome. Over time, her visual function improved where her side vision loss got better 3 months, and a year later was really all the way back to normal. We were able to watch this recovery over time in this unique condition and count the cones that appeared to be missing the first time we saw her when her vision was reduced, became much brighter and appeared to have returned by the time her vision had improved. So here's a movie that puts the images on top of each other. Here we saw at the beginning, very few of those cones look like they're present at all. But by a year later, when her vision had improved, we begin to see the cones return. So this,
Dr. Duncan, you're at
yes.
the 18-minute mark, so you should wrap up, please.
Perfect. I'm just at my last 2 slides. So thank you for that. This suggests that we can see the cones and we can monitor, identify the cause of the vision loss was reduced reflectivity of the cones, and we can monitor recovery over time. We can use this same approach in clinical trials. We've gotten funding from the National Eye Institute to deploy these systems at a number of different sites. We're going to be using a phantom that allows us to calibrate the images collected at different sites to see, to ensure that the images are comparable from different locations, using a nanoscale phantom that will be imaging at all the different sites. So to summarize, this is a new sensitive tool that allows us to study vision cell structure and function in a non-invasive, objective way. We can use it in combination with things like OCT and visual field testing to better understand the information we get from each of those other types of images. And the brightness of the cones is often reduced in patients who have vision loss, and that brightness might be something else we can track over time. And we think this is a new technique that will help us monitor disease progression and treatment response in patients with retinal degenerations. And these are the funding sources that have made the work possible. Happy to answer any questions. Thank you.
Thank you, Dr. Duncan. That was wonderful. You explained a lot of really complex issues there in simplistic terms for many to understand. Our second speaker, who I'm going to introduce to you now, Dr. Krzysztof Palczewski is currently Professor of Ophthalmology at the University of California, Irvine, serving as director of the Center for Translational Vision Research. He is a distinguished biochemical pharmacologist and molecular biologist, known particularly for seminal multidisciplinary scientific contributions to the biology and chemistry of vertebrate vision and in therapy for retinal diseases. His laboratory is best known for solving the structures of different forms of rhodopsin and other important proteins in the visual system. And they've also developed high-resolution imaging that impacted non-invasive in vivo monitoring of real-time visual function. Dr. Palczewski was born in Poland and achieved multiple degrees, including a PhD there, before moving to the USA, where he has worked at the University of Washington, Seattle, and was chair of pharmacology at Case Western Reserve University in Cleveland, Ohio. He's received numerous prestigious international awards and is a member of both the National Academy of Sciences and the National Academy of Medicine. Over to you, Dr. Palczewski.
Thank you very much. Good morning and good afternoon. It is a perfect time to present our ideas on treatment of inherited retinal diseases. And I haven't been more excited about research in terms of funding the cure for the blinding disease than recently with the support of the Stein Innovative Award to develop this technology. So we just about 5 years into this research projects, and I really would like to start with the very brave statement that I hope these 90,000 patients that are affected by inherited retinal diseases will have a chance to be cured and genetic native sequence can be restored. So let me start with a problem to present it to you. And I will start as an example of Leber congenital amaurosis, LCA2, the disease that has been described by Theodore Leber a long time ago. It is relatively rare disease, but of course, for those who are affected, it is 100% disease. It's very severe blindness, and there are 8 genes that were identified to cause the similar phenotype. The most common LCA is the most common cause of inherited blindness in childhood. It causes loss of function, mutation in genes that are important for key processes in the retina, obviously. And it took decades of developing both chemical pharmacological treatment of this one single disease. And also, this was the subject of the first FDA-approved gene therapy developed at University of Florida and Pennsylvania. So there was a progress, but again, it was a very long time before we had the fruits of this work. This again went through a pharmacological treatment through study in animal models, small rodents, as well as in larger animals like in dogs. But the success was truly remarkable with this approval of Luxturna in 2017. And this technology is based on a rather benign virus, AAV, that delivers a copy of proper sequence of an enzyme that is otherwise inactivated due to lesion in that particular gene called RPE65. So the virus, once it enters the cell, so it has to be injected into subretinal space. Once it's injected, it forms this episomal DNA. And for a relatively long time, perhaps a decade or so, it starts expressing the protein and the sequence, which is not a sequence of the patient, but rather improve and fix the sequence that otherwise this patient carries. This is example from biochemical point of view that is relatively easy to understand. You have a missing enzyme and you providing a copy of the DNA that can be translated into a protein that is functional. Much more complex problem arises when we're dealing with a structural protein, when the concentration of that protein will be critically important that matches the wild type expression of those particular genes. So the other complication, if you noted what I started with, that it takes decades to develop those type of technologies, that if we look through the retina, there is a large number of genes that are affected that cause similar phenotypes. So here is just a list of some of those genes. They are related to the mutation in, let's say, bestrophin, RP65, affecting RPE cells, but there are also mutation in genes that are expressed in photoreceptor cells. Perhaps the best study is ABCA4, which causes Stargardt disease, and rhodopsin mutation, and so on. So this is a large number of those genes that are affected that could lead to blindness. So if you also consider that one protein, and in this case is PDE6, that shows that there could be 100+ different mutation in that particular gene, and there can be stop codon, it can be missense mutation, it can be rearrangement and deletions. So there are many changes that can happen to PDE that will result in a particular phenotype, and not necessarily the same phenotype, depending on the mutation. So this is quite a complicated, and this is not even going into much more complex disease like age-related macular degeneration, when etiology is a little less clear than for inherited retina diseases. So clearly, if we wanted to take 10 years to develop a drug against one particular mutation, in some cases against one specific mutation in a specific gene, this will take forever. So these slides again illustrate that there are at least 250 genes where mutated, it leads to blindness. So we need to have some other approach that will enable us to address the issue of high heterogeneity and complexity of the blinding diseases. Seems to be hopeless, but just in the last decade, with combination of the progress in medicine, with this Luxturna and viral delivery, with understanding of the genetics of all of those human diseases, we're perhaps in time of dramatic changes of treating those diseases. As Dr. Duncan mentioned, you can observe those pathologies, and the patients, of course, have the reduced vision, but what can we do about it? So going back to genome, we have a chromosomal recombination between genetic material mom and dad. Each of us, dad and mom, contribute genetic material, and there are additional changes in the genome that are results of crossover between the chromosome coming from both parents. So that's why we become quite unique at the end. And there are diseases related to mutation in one just copy, and that copy is produced and causes trouble. And that will be autosomal dominant inheritance, that one copy that is carried by either affected father or mother will be transmitted to children. And here you will have frequency about 50%. The good example are mutation in rhodopsin that cause autosomal dominant inheritance of this disease. It's a progressive disease, and again, one copy is insufficient, or one copy is because of mutation causes abnormality on cellular level. Autosomal recessive will affect, in this again, statistical picture, just one child where both parents are the carriers, and the child will be affected because the gene will be inactivated. So this is a very simple background to what we're going to do. How about doing DNA engineering? How about if we go to DNA and try to modify that particular site that is not normal, it's affected by mutation? Can we do that? And recent progress in technology, development of CRISPR, really raised the hope that this will be technology that will enable us to do a DNA engineering. So instead of doing pharmaceutical addition or adding the virus, we will just simply go to the heart of the issue, the changes in the chromosome, the changes in DNA. And the first step in that was development of CRISPR that cut DNA in a specific site because it's guided by this green RNA. So it goes anywhere in the genome and can hybridize there and bringing the enzyme to cleave DNA, and in the presence of the donor template, the DNA can be repaired. This technology is, again, conceptually relatively simple, but in practice has many drawbacks, particularly that the recombination, it's at a very low level, on the order of 1% or less of the DNA will be fixed. So this is not really applicable as a method to make changes in this genome engineering, but rather it is a methodology that was waiting to be further developed. And one of the pioneers and our close collaborator on this project, David Liu, thought about this process in a slightly different way. He modified the enzyme involved in this DNA cleavage called Cas9 and make changes to it that it does cleave only one strand of DNA or doesn't cleave at all. And the RNA can deliver the specific complex of the Cas9 and RNA to specific site of DNA that the change can occur, because also Cas9 has been hooked with the enzyme called aminase, the aminase domain. So this is the base editing that is extremely efficient. It could be up to 80% of efficiency, where the changes in the genome can be achieved by this modification in a specific site. The drawback of this methodology is that not all of the mutation, and particularly when we have deletion, can be fixed by the base editing. So in parallel, the technology just in the last few years was developed called prime editing, where instead of the aminase, reverse transcriptase is connected to Cas9, and that combination will enable virtually any deletion and changes to be up to 4 kilobases to be repaired and fixed to a template that will provide a native sequence. I know that for some of you, this may be a little bit complicated with the jargons and different names, but in short, what the new technology of base editing and prime editing will do, will go to a specific site, any site that we design by using this very short RNA, that it can go to this site and fix the sequence that is responsible for pathology. It is extremely powerful methodology, and it's very powerful for several reasons, and namely that it will restore the sequence to the native, and it is also important that the drug can be delivered only for a very short period of time. So whatever design of the drug is, if we deliver this Cas9 with RNA to the cell, this change will happen, and the entire system, the Cas9 reverse transcriptase and the guide RNA can be destroyed. It's not needed anymore. It is just a simple, one single reaction that needs to take place. So our focus right now is on the delivery of the system to the eye that is transient, that is not causing less desirable changes in DNA, that it only acts for several hours and then is destroyed. And here's our system using Cree methodology, which is a homologue for this Cas9, where we have every cell in the retina expressing red fluorescent protein. And if we deliver this entire system to those cells, it will switch the color from red to green. And those are the two photon images that you can see on those slides, and that changes the color from red and green. So readout of these changes is extremely simple. It is changing of the color that we can count it. Of course, when we use Cas9, the way to do it, it will be to do sequencing that the particular sequence has been corrected. So this is another example of different methodology that we use to deliver. In this case, again, very many of the RPE cells are affected and they change the color. And also another way of delivering is to use a different system that enable us to go specifically to photoreceptor cells and to deliver DNA material or protein material to those cells to cause changes. So in advantage of the base and prime editing are very powerful and relatively simple. Namely that this methodology will enable to correct the genome under physiological condition, holding the expression of the mutant protein. So we'll address the issue of autosomal dominant and autosomal recessive diseases, because in case of autosomal dominant, the protein that is really a pathogen for the system now will be corrected to proper sequence. There are different types, of course, in the retina, and those cells can be targeted by different approaches to deliver this genetic engineering tools to make the changes in different type. This immune privilege of the eye enable to deliver proteins or RNA or any other things that is transient without really worry about the immune response, at least initially. But once again correction is made, the system will stay on in a precise native sequence. And vision is also going to spearhead these changes in genome editing and genome engineering, because we can measure precisely the outcome of our changes, not only by sequencing like in animal models, but also by visual performance in varieties of techniques that were developed in the last decade. And there are multiple advantages of delivering this protein from the point of view of pharmaceutical agent, because that delivery needs to be only transient, and it has to be, if enzyme is active enough, need to only go for several hours, and that should be sufficient to changes the DNA makeup of the target.
Dr. Pelczewski, you're at the 18-minute mark, and you should please wrap up.
And most also important feature of this system is that this genome editing can bring the changes specifically to any place in the chromosomes. So this is how that technology will look like. We will start with identification of mutation in the patients. Then there is a theoretical in silico design of this genome editing material or machinery. Then you will take the biopsy, the skin biopsy, develop those cells, and further select the material that will be used for in vivo study, packed it into a RNA or protein way of delivering, and inject it into eye to change the mutant into a native sequence. And again, very quickly, I wanted to thank people involved in this project, and really wonderful graduate student who graduated now, Susie Su, who was inspired by Nobel laureate Jennifer during our ARVO meeting in Hawaii. So this was a very important meeting for Susie to move entire lab now into a large part of the lab into a genome editing approach. So with that, I will thank you and take any questions as they come.
Thank you, Dr. Pelczewski. That was incredible. Such amazing futuristic work that you're doing. But let me start with this question. And I'll ask this to both of you, but let's start with you, Dr. Duncan. For people watching this seminar who have been diagnosed with retinitis pigmentosa, at least clinically, what advice do you have for them?
Well, I would say one thing that I think when people have just been diagnosed, it can be a little overwhelming to hear that you have a disease that is genetic and inherited and may or may not have, likely doesn't have an effective treatment at this time, and it may affect other family members. But what I want people to maybe understand is that this is a part of who they are. It's intrinsic to their DNA. They've probably had it their whole life, whether it caused them any trouble or not. And it likely will change over time very slowly. So the good news is there's a lot of exciting research underway. You heard about some of it today from Dr. Palczewski, and there's lots of treatments in development to slow down the rate of vision loss. This tends to be a disease that progresses very slowly. So people have a lot of time to develop support and get used to the idea of, they probably had been coping with vision challenges for many years. So the thing to remember is it's very slow. There's a lot of exciting work underway, and I'm very optimistic that there will be treatments in the not too distant future. In the meantime, you should seek out a low vision consultation to make sure that you have as much support as you need, whatever the vision challenges that you may be experiencing may be.
Thank you for that. Dr. Palczewski, do you have anything to add?
I think that the genetic testing right now is not prohibitively expensive, and it should be done. Because as these technologies are coming to potential testing, you wanted to know whether pharmacological or this DNA engineering, whether that will be an area where you could participate very early on in developing the treatment. So I think that, again, anybody with visual deficiency, if there is any chance to do DNA testing, would be highly preferable.
Right. So you'd encourage people to get genetically tested, know exactly what pathogenic variant they have. That'll help inform the type of retinitis pigmentosa that they have, and then they can go on from there, perhaps seek out if there's a clinical trial or something in the works for them. That's great. Thank you. A question from the audience has come in. Not sure who this is for, but let's. I
can take it if you wish.
You'll take it. You start it. Okay, you can start. Are there some rods more susceptible to degeneration in RP? Why are not all affected equally? And I think this might be because of the mid-peripheral loss that we see to start.
Yeah, so not all of the rods are the same, and there is a really geographic difference between very peripheral rods, those which are in the zone around the macula. They are even morphologically different. They more dense, they require more energy to be provided to them, and they are skinny and less healthy, maybe. Hard to tell, but there is again geographical difference. They are not one rod is the same throughout the entire retina.
Oh, that's great to know. Dr. Duncan, anything to add?
That's absolutely excellent explanation. The other thing is that I think the ones that are, there's some argument that exposure to light may contribute. And so the ones that are, the rods are densest, as Dr. Palczewski said, are along the arcades. And those are the ones that are sort of exposed to the light the most because they're in the macula, which is the part of the retina we use to look at things. So I think that in some ways, the highest density of rods that are kind of central are the ones that are most vulnerable for any number of reasons, including some of the ones Dr. Palczewski said.
Okay, that's really great. Excellent, excellent. This is a question specifically for you, Dr. Palczewski. Are the base and prime editing approaches being tested yet in patients, or are they still in the preclinical stages?
So this is still experimental treatment in animal models. So the whole technology based prime editing, for example, has been invented just 5 years ago. And right now, entire technology moved from cell lines into animals. So even in my lab, we're using 3 different models for RP to develop base editing and prime editing. And some of them, some of this work has been already published. So I think because of this progress with Luxturna and other ways of delivering DNA, RNA material to cells and patients that are right now, it is possible to accelerate that process going from the genetic information about the patients to designing the guide RNA to testing. I hope it will be much faster, and particularly after the first disease will be treated, I think that it will be a playbook how to go back to do a separate, different mutation corrections. That is going to be virtually the same material used. So I think that acceleration of this technology will be just incredible after the first step will be passed through and the first clinical trials will be conducted, because it is exactly the same material that is going to be used, except a very small piece of RNA that I hope FDA will have its own decision how to go about in terms of toxicology, whether each of the RNA will have to be retested and retested, or this become a playbook from one developed technology going forward.
Good to know, good to know. For you, Dr. Duncan, how common is the use of adaptive optics in clinical practice? Has it gone from the lab to some of the specialist centers now, or is it still a research tool?
Well, adaptive optics actually is largely still a research tool. There are some commercially available systems. So some retinal clinics around the country and around the world do have access to adaptive optics systems. The ones that that I described today are not that type. They're research tools. But our goal is to try and develop protocols that allow us to validate the information collected from different systems, even if they're not the exact same system. The same kind of information comes out of all of them. So by using this standard phantom where everyone takes a picture of the same thing, which is like it's got different spacing of cones between it, like a human person's eye might. We hope to be able to say, even though these systems aren't available at Kmart, whenever you can't buy them on the street, they provide the same kind of information that could be useful for patients, and certainly at the types of highly specialized centers that really are doing research to characterize these diseases. A number of them do have adaptive optics capability.
Okay, wonderful, wonderful. We've got a specific question here about X-linked RPGR gene defects. What are the chances a female with symptoms would require treatment? I guess a female carrier of X-linked RPGR. Dr. Duncan, you're nodding your head, so
Sure.
to you first.
That's a very interesting question. So the challenge has been female carriers who have mutations in genes that cause X-linked RP are very variably affected. So some patients are very severely affected, just as their sons and fathers might be. Others have no idea they have any problem at all. And sometimes it'll be in only one eye or one part of one eye. But it's the same process that causes vision loss in men. The challenge is that in women, it's a very patchy, sort of unpredictable expression. So the trials have largely focused on the men, demonstrating that a treatment works is easier in somebody who has more severe disease that is the same between both eyes. That being said, I think if a treatment is done in men and found to be safe and effective, women are very likely to benefit from that treatment. There are some trials, I think, that are beginning to consider enrollment of females, but please understand the complexity that introduces, because women are so different from even one eye to the next, even one region of one eye to the next. It makes it challenging for the clinical trial to be designed that is going to demonstrate a treatment is safe and effective in them.
Right, great. And Dr. Palczewski, you mentioned expression of RPE65 in Luxturna for 10 years or so. Is that just based on the data that's available now, or is that a prediction of durability?
No, we know that this is again extremely careful study done by Arthur Sidesian and Sam Jacobson, that even after treatment, the degeneration is still progressing. So the question, of course, that this raises, if the changes due to expression of RP65 are not permanent because already those cells trigger the death pathway that they're going to degenerate, or is it simply a lower expression level as we go over the time that, 10 years later, there's so much less of expression that causes the problem. Again, this required a pharmaceutical agent. It requires the amount of this viral particle pretty high. And so if the second injection is virtually not a possibility because of the activation of the immune system. And therefore, again, I will advocate for these permanent changes, for DNA engineering as a better method to do it, because you have many different ways of delivering the necessary material to those cells, and the permanent change that is going to take place after that mutation is fixed. It will be superior to, I believe, to the viral delivery as we know right now with AAV.
Good. And a question about how you go about treating a genetic disease that impacts both vision and hearing, for instance, Ushers. How would you treat that? I'll start with you, Dr. Duncan.
Yeah, that's a really important question. So some diseases cause congenital hearing, like Usher syndrome, causes congenital hearing loss that is present very, very early on and stationary. It doesn't get worse over time. Others cause some, for Usher syndrome type 3 and other kinds of syndromic diseases, cause progressive hearing loss. So there might be some opportunity to treat the ones that cause progressive hearing loss in the same way that we're treating progressive vision loss. The challenge is that delivery of the treatment in ophthalmology is something that people have been very eager to pursue, because the eye is localized and you can deliver the gene exactly where you want it to go, and it's not likely to be causing other problems elsewhere in the body that you don't anticipate. And so you may envision a scenario where the eye and the ear are both separately treated to replace the missing gene. This is in comparison to some syndromic diseases that affect the whole body. For example, Batten's disease, right? There are some diseases where there are systemic treatments being developed to try and address multiple complications that arise from a genetic change that also may affect the eyes and the ears. And that might require more of a widespread kind of a treatment approach. So it depends on the cause of the hearing and the vision loss, I would say.
Thank you so much.
Well, unfortunately, we're at the end of our time, and I just want to wrap up the session and be respectful of people's times. But this was really exciting stuff, and thank you so much, Dr. Hicks and Curran, for a great job of moderating. But also thank you, Dr. Duncan, for presenting really cutting edge research about the diagnosis of RP, and Dr. Palczewski, thank you so much for giving us a vision of what might be possible in the future in terms of treating these diseases with really very innovative gene editing techniques. So again, thank you all, and I hope that everyone has a good rest of their day. Thank you.